Carbohydrates are the most abundant organic compounds in nature. Now, carbohydrates are also known as sugars, and carbohydrates are polyhydroxy aldehydes or ketones. So that means that they contain multiple OH groups as well as an aldehyde or ketone functional group. So we'll see a lot of the chemical reactions that carbohydrates undergo are the same types of reactions that we saw in the aldehydes and ketones chapter. Now, the molecular formulas of sugars or carbohydrates are that they contain a specific ratio of carbon to H2O molecules. So this is where the term carbohydrate comes from. The carbo comes from carbon, and hydrate is another term that we use for water. Carbohydrates are naturally synthesized in nature through photosynthesis. So plants will take carbon dioxide as well as water and use sunlight to help turn it into a carbon dioxide known as a carbohydrate known as glucose, as well as producing oxygen. Now, glucose can then be turned into a variety of other carbohydrates. And ultimately, these carbohydrates are going to be used and synthesized by plants, animals, and other humans for a variety of reasons. So carbohydrates can be used for energy storage and other biological processes. In this chapter, we're mainly going to focus on the classification of carbohydrates, and we're going to start with classifying monosaccharides. So monosaccharide is a term for simple sugars or simple carbohydrates. And what that means is they cannot be hydrolyzed to other sugars. Later on, we'll see examples of non-monosaccharides, so things that are disaccharides, trisaccharides, or polysaccharides, and we'll see how those can be broken down into monosaccharide units. Now, these are examples of some monosaccharides. So we have glucose, which we just saw that plants can synthesize, as well as fructose. Now, carbohydrates are often drawn as Fischer projections. And if you recall from Fischer projections from Chem 226, any bonds that are in the horizontal position act as wedges. Anything that is vertical acts as a dash for the purposes of determining stereochemistry. Now, as we can see, many carbohydrates can contain several chiral centers. So for instance, glucose has a total of 1, 2, 3, 4 chiral centers, where fructose has 1, 2, 3 chiral centers. So in the first part of the classification of monosaccharides is we focus on the type of functional group that the carbohydrate contains, as well as the number of carbons. So we use the term aldol to focus on a carbohydrate that contains an aldehyde group, and this is going to use the prefix aldo. We use ketose to classify a carbohydrate that contains a ketone group, and we use the prefix keto. Then we use the terms di, tri, tech, pent, hex, hept to indicate the number of carbons. And these are the most common number of carbons that we're going to find in carbohydrates. So we have 2 through 7. So for instance, this carbohydrate here contains an aldehyde functional group and a total of 1, 2, 3, 4, 5 carbons. So this is known as an aldol pentose. So that ose indicates the suffix that we use for carbohydrates. For this one over here, we have a ketone functional group and a total of 6 carbons. So this is known as a ketohexose. Now, even though carbohydrates contain multiple chiral centers, for classifying chirality, we focus on the chiral center that's farthest from the carbonyl. And we are going to be using D and L designations to determine the stereochemistry. Now, D sugars, which are the most common and naturally occurring, have the OH group at the chiral center furthest from the carbonyl on the right-hand side. So all of these, D-ribose, D-glucose, D-fructose, focusing on this chiral center furthest from our carbonyl, have the OH on the right. Now, this is also going to correspond to the R configuration. We can go ahead and practice determining stereochemistry just to recap from Chem 226. So again, focusing on this chiral center, we're going to determine the priorities of the groups. Now we have an oxygen directly attached in our OH group. that's going to be priority number 1. Hydrogen's always priority number 4. So now we need to determine the priority of the other groups attached. So this carbon here is attached to an oxygen and 2 hydrogens, whereas this one here is attached to a hydrogen, carbon, and oxygen. So the total atomic numbers for this one on the top is higher than for the carbon on the bottom. So this is going to get priority number 2 and priority number 3. Now we learned that we trace a path from 1 to 2 to 3, and we are currently going in the counterclockwise direction, which would correspond to the S configuration. However, our 4th priority, which is a hydrogen, is on a horizontal line, which corresponds to a wedge bond. That means that we are looking at the molecule backwards. So in order to determine our R and S configuration, our 4th priority must always be a dash. So that means we're going to be using the opposite configuration for R. So that's just going to prove that our D sugars correspond to the R configuration. Now the reason that we use D and L designations rather than R and S in carbohydrates has to do with the specific rotation of the simplest carbohydrate, glyceraldehyde. So glyceraldehyde is the simplest aldol. It contains an aldehyde functional group and also 1 chiral center. Now, all carbohydrates can ultimately degrade down to glyceraldehyde. So that's why we focus on glyceraldehyde for these L and D designations. Now, the reason that we use D stands for dextrorotatory, which corresponds to the direction that glyceraldehyde rotates plane polarized light. And glyceraldehyde rotates that light in the positive direction. So positive corresponds to dextrorotatory. Now for its enantiomer, the L-glyceraldehyde, it's going to rotate the plane polarized light in the opposite directions, the levorotatory direction or counterclockwise direction. So with this one, we have the OH group on the left. So that's where these terms L and D come from. It comes from the direction that glyceraldehyde rotates plane polarized light. Now it's important to note that dextrorotatory versus levorotatory does not correspond to the D and L designations in most monosaccharides. So this D and L comes from glyceraldehyde's ability to rotate plane polarized light. However, in other monosaccharides, even though they're classified as D configurations because they have the OH group on the right, similar to glyceraldehyde, they're capable of rotating plane polarized light in either direction. So these are examples of 2 carbohydrates that even though they have the D configuration, they rotate light in the levorotatory direction or counterclockwise direction. So this D and L is just a classification for where the OH group is placed. So for our purposes, and this is really what you mainly need to remember, a D sugar means that the configuration of the furthest chiral center is R, whereas a L sugar has an S configuration. Or another way to think of it is a D sugar has the OH on the right, whereas an L sugar has the OH on the left. Now we learned in chapter 19 that an aldehyde can react with an alcohol to produce a hemiacetal intermediate under protic conditions. Now as we saw in this mechanism for acetal formation, the hemiacetal is actually highly unstable and the molecule would prefer to continue on in the mechanism to form a full acetal. So just forming an acetal is not favorable, so we would see the equilibrium favor just the reactants. However, when a hemiacetal formation occurs to form a 5 or 6 membered ring, the equilibrium does favor it. So for instance, if we have this intermolecular reaction with an aldehyde and an alcohol under acidic conditions, we can produce a stable cyclic hemiacetal that is capable of being isolated. So monosaccharides will react with themselves to undergo ring closing to form a hemiacetal. Now in this process, we do produce a new chiral center, and this is called the anomeric carbon, and this will always be at carbon number 1. So essentially, we take the Fisher projection and we can draw it in more of a ring structure. And we'll kind of practice drawing this ring on the next slide. But under these acidic conditions, we can have the aldehyde functional group as well as the alcohol come together to form our hemiacetal. So remember that a hemiacetal is defined as an OH group and an OR group attached to the same carbon. So not only do we form the hemiacetal on carbon number 1, but we also form a new chiral center. Now the equilibrium for this reaction greatly favors the cyclic form. And again, that's because we're forming those six-membered rings, which are the most stable rings possible. And our six-membered ring, hemiacetal, is also known as a pyranose ring. Now let's go through the process of drawing a cyclic monosaccharide from a Fisher projection. And these cyclic structures are known as Haworth projections. So first what we're going to do is we're going to take our Fisher projection and we're going to lie it down on one side. And this is going to help us know what direction the OH groups need to be pointing in our ultimate cyclic structure. So the OH groups that are on the bottom are going to be pointing down below the plane of the ring. The groups on the top will be pointing up above the plane of the ring. Okay, next what I'm going to do is I'm going to take all 6 carbons and I'll draw them in a cyclic form. So I'll start with carbon number 1 on the right. And carbon number 1 contains our aldehyde functional group. Then I'm going to go down, I'm going to go clockwise. I have carbon 2, 3, 4, 5, and then carbon 6. I'm going to go ahead and draw CH2OH. Now I'll go ahead and fill in the H's and OH's. So on carbon number 2, I have an H going up and an OH pointing down. On carbon 3, I have an OH going up, an H going down. Carbon 4, I have an OH going down, an H going up. On carbon 5, the H going up and the red OH pointing down. Now the red OH is ultimately going to be forming the bond between carbon 1 and itself to form our hemiacetal. So to kind of set this up, I'm going to rotate the groups attached to carbon number 5 around. So I'm going to shift over the OH group into the place of the CH2OH. I'm going to shift this over to the H, and the H is going to go down to the OH group. This will make sure that we keep the stereochemistry exactly as it should be on carbon number 5. We're just switching the places of all 3 groups. So now on carbon number 5, just this one here, we have CH2OH, we have our H pointing down, and then we have our red OH pointing over. So now we're all set up for the hemiacetal formation between this OH group and our carbonyl. So the mechanism is exactly the same as what we've learned for the hemiacetal formation. We protonate this oxygen, the red one, comes in and attacks, kicks the electrons up, and then ultimately we are going to get rid of one of the extra hydrogens. Now we are going to form a ring as our product as well as a new chiral center. I'll just leave out the H's for now. But on carbon 1, we're going to have a new OH group. Now the OH can either be pointing up or down. I'll go ahead and draw it pointing up right now. But this one can be up or down. And depending on whether it's up or down, is going to form a type of stereoisomer known as an anomer. We'll take a look at what anomers are on the next slide. Now we know that the OH on our new chiral center can form below the plane of the ring or above the plane of the ring. And actually both of these will form to make stereoisomers. Now these stereoisomers are known as anomers. And they are given designations as alpha or beta depending on where this OH group is in respect to the CH2OH group of carbon number 6. So the alpha anomer is when we have the OH group trans to the CH2OH group. So this is the alpha anomer here. This group is below the plane of the ring. This is above the plane of the ring. So that makes them trans with one another. Beta is when the anomeric OH group is cis to the CH2OH group. So in this case, they're both above the plane of the ring, which will make them the beta anomer. Now, it's not always possible to predict which one is going to be the major product, so it's typical just to draw both unless you're asked to draw a specific one. So pyranose rings are typically drawn as Haworth projections, which are these ones here in more of a hexagon shape, or in chair conformations, which we've learned back in Chem 226. Now the chair conformation is better, it's more accurate because it reveals the 3D conformation. Now, whenever you're taking a Haworth projection and drawing it as a chair conformation, we always put the oxygen drawn in the upper right back position. So this is how you're typically going to see the chairs drawn for our carbohydrates. Now, this is the chair conformation for glucose. And it's important to note that glucose is the only sugar that's capable of having all of the groups equatorial. So it is the most stable sugar. There are no groups in the axial position, which is why glucose is the most abundant carbohydrate in nature. So where a pyranose ring is a 6-membered ring hemiacetal, a furanose ring is a 5-membered ring hemiacetal. Now fructose, which is this carbohydrate here, it has a ketone functional group. So this is a ketohexose. Now, fructose is capable of forming either a 5-membered ring or 6-membered ring, depending on which OH group is going to act as the nucleophile to form the hemiacetal. So in the furanose ring formation, we have the OH group attached to carbon number 5 doing the attacking. Now, ultimately, that's going to form our 5-membered ring. In the pyranose ring, we have the OH group on carbon number 6 doing the attacking. Ultimately, that will come in, form a 6-membered ring. So fructose is capable of doing both of these and forming both versions, the furanose and pyranose rings. So as we saw in chapter 19, hemiacetals can continue to react to form acetals. So we can see the same things with sugars. A cyclic hemiacetal of a sugar can continue to react with an alcohol under acidic conditions to form an acetal. So again, this is just exactly the same as what we saw in chapter 19. Now, acetals of sugars are specifically called glycosides. So what we can do is we can take the form of a molecule or carbohydrate that is a hemiacetal, continue to react it, and turn this OH group, which makes this molecule a hemiacetal, into an OR group. So now it is an acetal. Now again, these groups can form either below the plane of the ring or trans to our CH2OH group, or it can form on the same side of the ring or cis to it. So you can get a mixture of both the alpha and beta glycosides, similar to forming the alpha and beta hemiacetal sugars. So the mechanism for glycoside or acetal formation is similar to the formation of a typical acetal that we saw in chapter 19. So again, we have the OH group that's going to be doing the reacting. We're going to protonate the OH group with an acid and turn it into a good leaving group, that is water. Now, either the water can just leave to form a resonance stabilized carbocation, or sometimes we can draw it as showing the oxygen donating its electrons and kicking off the water. Either one of those is appropriate, but either way, we're going to form a resonance stabilized carbocation. Once we've kicked off the water and formed our resonance stabilized carbocation, the alcohol can come in and perform a nucleophilic attack. Now, depending on what side of the molecule the attack occurs, that's going to determine whether or not we produce the alpha or the beta glycoside. So if our alcohol attacks from the top or the same side as our CH2OH group, ultimately it's going to attack, kick the electrons up onto oxygen, and then the final removal of the extra proton will produce our alpha glycoside. Alternatively, it can attack from underneath the opposite side of our CH2OH group, and then again a proton transfer to form our beta glycoside. Other reactions that monosaccharides can undergo is epimerization. So this occurs under strongly basic conditions, and the epimerization occurs on the alpha carbon to the aldehyde or ketone functional group. Now this is going to occur through a process that's very similar to tautomerization. So what happens is we take our molecule that has our alpha carbon and we turn it into an ene diol. Okay, so this is similar to the enol that we saw in the tautomerization. Now at this point, we have turned this chiral carbon, which is sp3 hybridized, into an sp2 hybridized carbon. So now it's trigonal planar and it has no three dimensions around it. Then this ene diol can tautomerize again back to its keto or aldol form. And in this process, the OH group can end up either on the right or on the left. And this is going to produce molecules that are known as epimers. So epimers are diastereomers that differ in configuration of only one chiral center. So here we have formed D-glucose and D-mannose, which are two different carbohydrates. As we can see, the last three chiral centers for these carbohydrates are exactly the same. They only differ in one of these chiral centers. So that is our definition of an epimer. It's a very specific type of diastereomer. So oxidation tests can be performed to detect the presence of reducing sugars versus non-reducing sugars. So an example of oxidation test could be the tolerance test. So we use the tolerance test in lab to determine if we had aldehydes or ketones. The aldehydes were ultimately oxidized, which would give a positive tolerance test. Now, all of these can be used to detect for the presence of reducing sugars. Now, the main way to tell whether or not a sugar can be a reducing sugar or not is to look at the type of functional group that's in the pyranose or furanose rings. Now, if the carbohydrates contain hemiacetal functional groups, hemiacetals, whether they came from a ketone or an aldehyde, are capable of being oxidized and are classified as reducing sugars. Glycosides, which are acetal functional groups, are not reducing sugars and therefore are not oxidized in these tests. So we've talked a lot about monosaccharides, which are the simplest sugars. Disaccharides consist of two sugars that are connected through glycosidic linkage. So essentially what's going to occur here is instead of having acetal formation occur with alcohols on carbon number 1, the nucleophile in the acetal formation is actually going to be the alcohol of an entirely other sugar. So in this case, we have glycosidic oxygen that's connecting carbon 1 of one of the monosaccharides to carbon 4 of the other saccharide. So this is called a 1,4 disaccharide because we're connecting carbon 1 of one monosaccharide and carbon 4 of the other. Now this specifically is called maltose, and this is a 1,4 alpha glycoside. Okay, and that's because this entire group is on the opposite side of our CH2OH groups. These make this an alpha linkage. Now maltose, which is this disaccharide here, is a reducing sugar because it contains a hemiacetal. So even though we have formed an acetal with this monosaccharide, this one still contains a hemiacetal. So that's going to make it also a reducing sugar. Now in disaccharide formation, whether we form an alpha or beta glycosidic linkage can actually cause us to form different types of molecules. So maltose, which we've just been talking about, contains a 1,4 alpha glycosidic linkage. Okay, so this makes a specific type of disaccharide. Now, if we were to form the beta glycosidic linkage between the same two molecules, it would actually form a completely different structure of cellobiose. So these two carbohydrates have different functions. It's similar to enantiomers of molecules also behaving differently. So this is just an example of another type of disaccharide. So lactose, which is the carbohydrate that's commonly talked about in milk, is also a disaccharide. It's made up of galactose and glucose linked together with a 1,4 beta glycosidic linkage. Sucrose, which is table sugar found in every kitchen, is also a disaccharide. It is made up of glucose in its alpha pyranose form. So that's this one here, its six-membered ring, and fructose in its beta furanose forms. That's where we have the five-membered ring. Now what's interesting is honeybees are capable of taking sucrose and converting it into a mixture of sucrose, fructose, and glucose. So we can either keep it as its whole form, or honeybees can actually split this up. Now these two are linked together by carbon 1 on glucose to carbon 2 on fructose. So this is a 1,2 glycosidic linkage. Now it's interesting to note that sucrose is not a reducing sugar. That means it does not contain any hemiacetal functional groups. So if we focus on the carbons that are capable of being hemiacetals, we'll see that the oxygens are attached to R groups. So in carbon 1, we have it attached to an oxygen, attached to an R group, and an oxygen attached to an R group. So this is an acetal functional group. Carbon 2 is attached to an oxygen attached to an R group and an oxygen attached to an R group. So again, we have an acetal functional group. So we only have acetal functional groups, which makes this entire molecule not a reducing sugar. Next, we have polysaccharides. So polysaccharides is when you have many monosaccharides connected to one another. And the most common example of a polysaccharide is cellulose. So cellulose is made up of several glucose units, 7,000 to 12,000 to be exact, and they are all connected through beta glycosidic linkages between carbons 1 and carbon 4. So this is an example of how cellulose is put together, but you have to imagine that there's 7,000 to 12,000 of these units repeating. Now cellulose strongly hydrogen bond with each other, and cellulose is the main structural component of plants. So this is what gives wood and plant material their super rigid structure, is all of these linkages. Finally, we can also form N-glycosides. So this is when we take the hemiacetal, treat it under acidic conditions, and instead of having an alcohol act as the nucleophile and attacking that double bond that forms, we have an amine instead. So this will form a product that's called an N-glycoside, and again, depending on where the N attaches, it's going to be alpha or beta. Now this is very important in DNA because the nitrogenous base pairs in DNA are connected to deoxyribose units via these N-glycosidic linkages. Ultimately, these are forming the nucleosides. So this is an example of a nucleoside in RNA. So we are reacting ribose, which is a carbohydrate, with a nitrogen-containing ring. So this is our nitrogen heterocycle or base. So this nitrogen here is acting as the nucleophile to form our N-glycoside. Same thing for DNA. We use a different carbohydrate, which is deoxyribose, and we're still reacting it with nitrogens in a ring, and again forming this N-glycosidic linkage. In DNA, the nucleosides are attached to phosphate groups, which ultimately form nucleotides. Then, these can all be connected to one another via the phosphate groups to form DNA strands. So these are really the building blocks of DNA. So the main thing I want you to take away from this video and this chapter is I'd like you to be able to classify a carbohydrate, so either as an aldo or keto, the number of carbons, and the D and L configuration. I want you to be able to draw Hayworth projections from Fisher projections, as well as the corresponding chair conformations. Be able to distinguish between alpha or beta anomers, recognize reducing versus non-reducing sugars, and have a basic understanding of glycoside formation.